Okay, I'm still admitting, but those of you who already in this meeting room, you know the drill about your camera. And Hazel, the cat is quite funny. Quite funny cat. Okay, you can see uh, at the top left corner of the Zoom video, it says live on YouTube, because it is live on YouTube. So later, after I check your attendance, you can actually continue this participation of yours. I mean, if there is a problem with the Zoom meeting, the connection of your Zoom at your place, then as alternative, we got this meeting on YouTube. Okay, but I'm not using the biomolecule one. I'm not using the account biomolecule, but I'm using the uh, Roberto Santanamola account. Okay, Roberto Santanamola. That's my name, and that's the name of the channel, Roberto Santanamola. In case you want to check that out. So it's already 10:02. While there are 27 of us here, 27 including Miss Leonita and two Robertos. Okay, there are two Robertos. As you can see, one is biomolecule and the other one is Roberto, WK8E, yeah? So that means if it's now 28, that means there are only 25 of you. 25 of you, well, there are supposed to be 27 of you, so let's check it out. Check this out. sounds, oh, how I may lie. Puchai, Shefferly hasn't come. Oh, Liu Qi Pa Jiu 十、11、12、13、14、15、16、17、18、19、20、21、22、23。Nathaniel, not yet come. Nathaniel and Ryzen, you need to rename. You need to rename yourself because it's not in accordance with what instructed by the school. Ryzen, you need to rename yourself. And I noticed that Lionel is still got your camera off. Yep, it's on now. Thank you. Arigato gozaimasu. Mongo mas. Arigato gozaimasu. Mongo mas. 28. If we are all here, then there's supposed to be 30. Okay, so two are not are still out there beneath the pale moonlight. Who the two are Shefferly and 20. I forgot 23. Nathaniel and Shefferly haven't come. Okay, 10:05. Then I have to start, no matter what. Guys, good morning. Before I start again, I'm going to check the cameras of yours. Like, for example, Mr. David, you have to lower your camera a little bit. Miss Francisca, you also have to lower your camera a little bit. Yeah, again, guys, I don't want to see your ceilings. I don't want to see the roof of yours. I don't want to see the headberg. If there is an iceberg, which is the tip of the ice in the ocean. So is it wrong if I call the tip of your head as headberg? So I don't want to see any headbergs. Okay, I don't want to see any ceilings. Okay, awesome, great. So first agenda I'm going to talk about today, which is, guys, you know already that today is the deadline for your project report. Okay, today is the deadline. Today, midnight. Shefferly is coming to town. I'm admitting Shefferly. Okay, so it's just Mr. Nathaniel now. So today is the deadline. 23, 59, 59, 59, 59, 59, 59. And I really hope that you can submit your report by the deadline. Okay, hopefully like that. Nobody turn in the assignment late. That's what I hope. And yes, there were some groups from some classes which sent me the report of theirs. I've checked out some, but some others haven't been checked out. So if you already sent me your report, project report, but I haven't over, of course, if you need to go to toilet, you may go to toilet. I will not forbid you from going to toilet because it's something important to be positively responded. Okay, now again, go back to the project report. So some already checked out, but some others haven't. If then during the waiting for the deadline today for you, I managed to check out yours, then I guess, oh, Mr. Ken is coming to town. Then you can receive back your report and do what you need to do about it. But if until the deadline I haven't checked yours due to so many things, maybe, hopefully no, not, then just submit the report. Okay, just submit the report. So if there's something that you need to change or modify or improve or whatever, things like that, I'm going to communicate with you via Google Classroom. Hopefully you understand this. So again, I'll be waiting for your project report today.
Suaranya ada dua.
Mine is my voice.
Yes, sir.
Wait.
Eh,
No, the other me not connected with the audio. So is it still like that? Dual voice of mine. Is it still like that, guys? Still like that or no?
enggak, sir.
Uh-huh.
Tadi nggak tahu. Kayaknya laptop saya.
Ryzen. Okay, so now that was the first agenda. Any comments? Any questions? Any question? No? Then I'm going to go to the next agenda today. As you can read on Schola, today's activities titled project report submission, if I'm not mistaken, and diffusion and osmosis. The last two items are new terminologies which are quite important in learning not only biology but also chemistry, okay, and maybe physics. Okay, so diffusion and osmosis, those are the two terms we are going to understand today. And I suggest that you pay attention on everything I'm trying to explain. There will be new terms coming up. So it's better for you if you hold a pen or a pencil or whatever writing utensils you can use. You like, go ahead, so that whenever there is a new term, you can instantly write it down. Okay, including you can also actually, since this is a live streaming on YouTube, then yes, I guess you can check out the recording on my YouTube, Roberto Santana Mola, later to get what you missed today. Okay, that can do, I guess. So let's start. Um, Talking about diffusion and osmosis, the two important things we are going to talk about, we are going to discuss, and we are going to learn today. Um, I've prepared a PowerPoint file to show you. Okay, allow me to share my screen. I'm sharing my screen. I have to connect the audio, make it optimized, and here it is, the PowerPoint file. Okay, diffusion and osmosis. At the end of today's activity, I am giving you an assignment. Okay, then I better show you this one first. Let me see, Google Chrome. Yeah, this is my YouTube. Sharing my screen. I have to connect the audio,
whatever.
make it optimized, and here it is, the PowerPoint file. Okay, diffusion and osmosis.
This step.
Now, if I go to your class on Google Classroom, here is your class, and I open the classwork, you will see that here you got collaboration project reports. This is where you are going to submit your report, and it's due today, tonight at midnight, 11:59, and at 10:30, which is about 16 minutes ahead. Not ahead, but 60 minutes, in about 16 minutes, 16 minutes, it's scheduled, it will be open, the bag in which you will submit today's assignment. What will be the assignment? You are going to make a writing. You are going to write a simple writing. Okay, what will be the contents of your writing? It will contain, your writing will contain of 4 items. The 4 items are: first, the definitions of diffusion and osmosis; second, the examples of each diffusion and osmosis; third, the method, the practical activity, the experiment we can use to learn about diffusion; fourth would be the simple experiment, the method, the procedure of learning osmosis. Okay, so let's go to the PowerPoint file. So I'm going to play this. Okay, this file contains 2 videos explaining the 2 terms. I'm going to go to the next slide, which got the first video. Watch carefully.
Molecules dissolved in a solution are in constant random motion due to their kinetic energy. One result of this motion is that dissolved molecules become evenly distributed throughout the solution. This tendency of molecules to spread out is an example of diffusion. But how do these molecules come to be evenly distributed? Let's start with a beaker of plain water. What will happen if we now add a lump of sugar to the water? A lump of sugar is composed of many individual sugar molecules, and even as a solid lump, the individual sugar molecules are in motion. When the lump is dropped into the water, it begins to dissolve. Individual sugar molecules move randomly and constantly from the area where they are common to the area where they are scarce. This type of motion, when molecules move from areas of their higher concentration to areas of their lower concentration, is called diffusion. Diffusion continues until all the sugar molecules become evenly dispersed throughout the beaker. The rate of diffusion is affected by temperature, size of molecules, and the steepness of the concentration gradient. Although not specifically shown in this animation, this is one of the processes whereby materials are exchanged between a cell and its environment.
Okay, that was the first video explaining about what diffusion is. And there are several new terms. As you can see on the screen, on your screen, on our screen, we can see that there's a beaker filled up with water, and into the water put in a lump of sugar. And actually you can try this by yourself at home. I mean, if you have a sugar cube, for example, you can prepare a glass of water and then you drop that lump of sugar into the water. You do nothing else. You don't stir it. You don't shake the water. You don't do anything else. You just wait and see. Okay? Slowly but sure, eventually you will watch that the lump of sugar becomes smaller and smaller and smaller, and then eventually it will be gone. It will be gone. And what exactly happened at that time is this process, diffusion. So diffusion defined as the movement of particles or substances or molecules, movement of particles, substances, or molecules down a concentration gradient. Movement of particles down a concentration gradient. So here comes another new term, concentration gradient. Okay, concentration means konsentrasi. Ya, itu tidak begitu menjawab pertanyaannya. What is the meaning of concentration? Konsentrasi. What is the meaning of simulasi? Yeah, it doesn't really answer the question. But concentration means the amount of something. Okay, the amount of something in a particular, let's say, area or volume. That's what concentration means. Okay, concentration means the amount of something in a given area or volume. Okay, for example, let's say that you got two glasses of water. Both filled up with the same amount of water. Let's say it's 150 milliliters of water. Okay, doesn't matter. But both glasses contain the same amount of water. And then into the first glass, you pour 1 teaspoon of sugar. 1 sendok teh gula di gelas pertama. Into the second glass, you pour, let's say, 3 tablespoons of sugar. 3 sendok makan gula. Okay? Airnya jumlahnya sama di dalam dalam masing-masing gelas, tapi di gelas yang pertama kamu taruh 1 sendok teh gula, di gelas yang kedua kamu taruh 3 sendok makan gula. Okay, you know immediately that the amount of sugar in the first glass and in the second glass is different, right? The amount is different. This also say that the concentration of sugar, because in the first glass you got now a sugar solution, in the second glass you also got a sugar solution but different amount of sugar. So the concentration of sugar in the first glass is lower than the concentration of sugar in the second glass. Do you understand that, guys? So again, that means the amount of sugar in glass 1 is less than the amount of sugar in glass 2. That's what concentration is. Okay, so the term was just now concentration gradient. Gradient, G-R-A-D-I-E-N-T, gradient. Gradien konsentrasi. What's that supposed to mean? Concentration gradient means perbedaan konsentrasi. You can see obviously that the concentration of glass 1 is certain and glass 2 is certain, and they are absolutely different. Okay, if you notice in the video, it said that there are 3 factors that determine the uh, diffusion. Okay, first, temperature. Okay, so if you put the lump of sugar in a glass filled with tap water, okay, the time the lump of sugar disappear could be different if the water in the glass is warm water or hot water or cold water. Okay, temperature, that's the first factor. The second one is the size of the molecules. And then the third one is the steepness. The video said steepness of concentration. Steepness artinya steep itu kan curam ya. So if the difference of the concentration is so very significant, so steep, then the speed or the rate of diffusion would be faster. Okay, because diffusion will happen automatically whenever there is concentration gradient. Guys, until here, can you follow me? Can you follow my explanation? Would you please open up your microphone and tell me whether you can follow me or not? Can you follow me, guys?
Yes.
Yes? Others? Hello, can you follow my explanation?
Yes.
Oh, thank you. Thank you very much. Thank God for that. So that's diffusion. Difusi adalah perpindahan zat atau molekul atau partikel dari tempat berkonsentrasi tinggi ke tempat berkonsentrasi rendah. Oke, itu difusi. Now let's get on to the next terminology. Okay, I'm going to go to the next slide. Okay, so you see that there's something different. The beaker in this slide got something in it, which is a membrane that separates the volume of the beaker into 2 different parts. Okay, one on the right, one on the left, while before there's no membrane. Okay, this one we will talk about osmosis. Let's watch this one.
Diffusion is the net movement of molecules down a concentration gradient. This process allows small molecules, such as oxygen and carbon dioxide, to cross the plasma membrane. Most polar molecules, such as sugars and proteins, cannot freely cross this lipid membrane. Although water molecules are polar, they are small enough to pass through the membrane freely. This special case of diffusion that involves the movement of water molecules across a membrane is called osmosis. If a molecule, such as urea, is added to one side of a membrane, it will not be able to diffuse across the membrane because it is both large and polar. Because of its polar nature, it will interact with other polar molecules such as the water. This interaction reduces the number of free water molecules on the right-hand side. With fewer free water molecules on the right-hand side, there is now a net movement of water molecules down their concentration gradient to the side with the urea molecules. Because more water molecules are moving into this area than are leaving, the water level on the right side will rise. If the osmotic concentrations of 2 solutions are equal, the solutions are isotonic. However, when the solutions have unequal osmotic concentrations, the solution with the higher concentration of solutes is hypertonic, and the solution with the lower concentration of solutes is hypotonic.
Okay, that's all. That's a plane just passed. Okay, so that was about osmosis. There is a definition I like the most out of numerous definition of osmosis, and this is in the definition is in bahasa Indonesia. Okay, osmosis adalah difusi air melalui membran semi-permeable. Osmosis adalah difusi air melalui membran semi-permeable. Just now we already talked about diffusion, and we've tried to already understood the definition of the meaning of diffusion, right? So osmosis is diffusion of water through semi-permeable membrane. Okay, let's talk about this definition part by part. Diffusion of water. This part tells us that whenever we are talking about osmosis, we are going to talk about the movement of water. We are going to focus on the water. We are going to concentrate on the water. Water. Okay. Once again, kapanpun, setiap saat, baik bila manapun kita ngomong soal osmosis, maka kita akan berbicara tentang perpindahan air. Yang menjadi fokus kita adalah air. Yang menjadi titik perhatian kita adalah air. Itu, tolong diingat-ingat. Waktu kita ngomong soal osmosis, kita bicara tentang perpindahan air. Apakah itu larutan gula, apakah itu larutan garam, apakah itu larutan kecap, apakah itu larutan seblak, whatever. Tapi ketika bicara tentang osmosis, berarti kita sedang bicarakan tentang pergerakan air. Okay, diffusion is the movement. Osmosis is diffusion of water. Osmosis adalah difusi air. Okay, melalui, through or across a semi-permeable membrane. This definition tells us that the presence of a semi-permeable membrane is a must, is mandatory. Okay? Jadi kalau bicara tentang osmosis itu harus ada membran. Membrannya yang sifatnya semi-permeable. Jadi kalau saya pegang plastik mika misalnya, ya, sebagai saya taruh plastik mika di dalam beaker gelas ini, maka air nggak akan bisa tembus dari kanan ke kiri. Itu namanya impermeable. Impermeable, cannot be permeated, cannot be passed through. Oke. Tapi kalau kemudian saya taruh misalnya kasa yang lubangnya gede-gede, ya, maka waktu saya taruh sebagai pembatas, semua bisa lewat karena lubangnya gede-gede. But semi-permeable membrane means that this membrane itu lapisan atau selaput, ya, this permeable membrane can only be passed through by certain particles or molecules or substances. Tidak semua bisa melewat membran ini. Semi-permeable. Jadi dia selektif. Bahasa Indonesianya adalah membran semi-permeable atau membran permeable selektif. Yang bisa lewat itu cuman yang tertentu aja. Oke? Itu definisi membran semi-permeable. Okay, that explain why on the screen you can see those three round green balls there, right? And you watch the video and you knew that those are urea, okay? Itu molekul besar, and it stays there on the right side. It cannot go to the left. Why? Because they are too big. They are too large. They cannot pass through the semi-permeable membrane. So again, let's try to conclude. Osmosis adalah difusi air melalui membran semi-permeable. Oke, berarti sekali lagi kalau bicara tentang osmosis, kita akan fokus pada air karena pergerakan air lah yang akan menjadi fokus kita. Oke, dan dia akan melewati membran semi-permeable, suatu jenis membran yang tidak bisa dilewati oleh gini, salah saya ngomongnya. Suatu jenis membran yang hanya dapat dilewati oleh partikel atau molekul atau senyawa tertentu saja. And that characteristics of membrane caused us to be able to see this state of beaker contents, right? See that the one on the right, the surface is higher than the one on the left. It's not a miracle. It's natural. It's scientific. Okay, so that's osmosis. That's osmosis. Guys, actually, what I explained just now, the definition of diffusion, definition of osmosis are among the things you have to include in your writing, yeah, as your assignment today. Any question until here? Is there any question? Any question? You see on your screen also that there are two terms, hypotonic, hypertonic. Tonic talks about the concentration. Hypo means lower, ya, konsentrasinya lebih rendah. Okay, hyper means higher, berarti konsentrasinya lebih tinggi. Kayak apa namanya, hipotensi, tekanan darah rendah, hipertensi, tekanan darah tinggi. Hipo artinya lebih rendah, hiper artinya lebih tinggi. Okay, yeah, Raisen. So, any question, guys? If there is no question, really, are you sure? No question? Then I'm going to spotlight my other me. Okay, I'm going to spotlight my other me, which is this one. Guys, this is also among the items you have to include in your assignment today, which is to explain how you would study diffusion. Okay, I got here. Okay, a graph paper. I got here a graph paper, atau kalau di bahasa Indonesia mah kita kenal itu dipakai di sekolah adalah, kelihatan nggak ya? Mana? Nah, milimeter block. Tah, ya milimeter block lah. Kita mah nyebutnya ya buku milimeter block gitu ya, bukan buku kotak-kotak. Milimeter block. I got graph paper over here. Okay, and you know that on a graph paper you have this larger squares, which area is 1 centimeters square. Okay, 1 centimeter square, and inside this 1 centimeter square you got 100 more smaller squares, which area is 1 millimeter square, right? So this is a millimeter block. Why am I using this? Because if I do this, when I do this experiment using graph paper, I will be able to measure what I want to measure later, okay? Because I am going to observe and learn about diffusion here, right? So you will see later. Now, graph paper, this is a Petri dish. Petri dish, okay? Petri dish, cawan Petri, okay? And you can also see that on the graph paper, I made a dot over there at the center. It's on the paper, okay? And I tried to position the dot right at the center of the Petri dish, okay? And beside all this, 1, 2, and water. I put water already in the Petri dish, okay? I also use this. Can you see that? Wrong. There. Yeah, wrong. Koe-poe. Gimana sih? Nah, whoa. How should I show you? Kupu-kupu. Kupu-kupu. Ini adalah food coloring. Okay, ini adalah food coloring. Food coloring. Okay, I also use spray bottle. Cannot be seen because it's too big, but I can show you here on biomolecule spray water. This is what I used when I poured the water into the Petri dish. So what I am doing now is I'm going to drop this food coloring. I'm opening the bottle first. Okay, I'm opening the bottle and I'm going to pour this ink here into the Petri dish. What I need to do is to make sure that I do my best to drop only one drop of the ink, the food coloring, right on the dot here, right at the center. That's what I'm trying to do, okay? Let's see if I can do this successfully. Cross my finger. Come on. Oh, okay. See, you see that the drop of ink is now spreading out. Is spreading out. Okay. Okay. Let me ask Mr. Brandon then. Mr. Brandon, would you please open up your microphone?
Yes, Mister.
Okay. Predict what will happen, Mr. Brandon. Ramal. Ini kira-kira apa yang terjadi, Mr. Brandon? What will happen?
Memudar.
Memudar. What? You mean the color will fade?
Yeah.
Oh, okay. And then what else? Is that? Okay. Thank you, Mr. Brandon.
Only that, I think.
Only that. Okay. Thank you, Mr. Brandon. How about Miss Francisca? Miss Francisca, do you have any further analysis or prediction? What will happen here?
Nyebar.
Pardon? Sorry?
Tintanya nyebar.
Menyebar. Okay, good. Thank you, Miss Francisca. Thank you, Mr. Brandon. Thank you, Miss Francisca. Miss Francisca said that it will spread out. Okay, it will spread out. And yes, both predictions are correct. I agree with both of you, and I believe most of us agree on that. As you can see, before I tried to drop the food coloring right at the dot. Okay, here at the center of the Petri dish. But now it's already this big. Using graph paper enable me to measure the coverage, okay, coverage. Seberapa banyak menutup, seberapa luas menutup of the food coloring by counting the squares. Let's see, ER, Sanse, Olio, Ci, Pa, Cio. Udah 9 cm, 10 utuh, 10 cm persegi ditambah ya hampir 12, 13, 14, ya kurang lebih 15 lah, 15-16 cm persegi. That's my guess because I haven't done any accurate counting of the squares, but later I can do that. I can find out how large is the coverage. Now I want you to notice something else further. What's that? Look, that at the center the redness is so dark. The red color is so red, while outside at the peripheral the redness is not as dark as inside, right? Guys, this shows us concentration of the food coloring. At the center, the place where I drop the coloring, the food coloring, the ink is highly concentrated. As it diffuses out, the outside part, the peripheral, the perimeter, the concentration is not as high as at the center. Can you notice that? See? Outside, it's less red compared to inside. So we are talking about the concentration here. And once again, I want to emphasize that what happening here is diffusion. Diffusion. The question is, let's see, Mr. Aaron, until when will this process, diffusion, happen? Do you think? Mr. Aaron? Do you have any prediction of how long it will happen? It will happen?
I think until all of it got covered.
Yeah, awesome. That's an awesome answer, guys. That's an awesome answer, Mr. Aaron. Guys, I want to underline here that whenever there is concentration gradient, there will be movement of particles. Setiap kali, kapanpun ada perbedaan konsentrasi, pergerakan itu pasti akan terjadi. Otomatis. And as you can see, this part at the center is so dark, means the concentration is so very high. At the peripheral, it's not as red as inside. means the concentration is lower. And here, even here, guys, you see, it's not red at all. Means the concentration of the food coloring is 0. So you see, there is a concentration gradient here. And the concentration gradient will trigger the movement of the food coloring. It will continue to move until, like Mr. Aaron said, until the ink is evenly distributed. It's all over the Petri dish. Great answer, Mr. Aaron. Great answer. And I believe all of you, all of the others also got the same answer for my question just now. So guys, do you understand this now? This is diffusion. Inilah difusi. Difusi terjadi karena perbedaan konsentrasi. Tinta yang tadinya diteteskan di titik di tengah-tengah cawan Petri, itu kemudian menyebar ke tempat di mana tinta itu konsentrasinya rendah, bahkan 0. Dia akan terus bergerak sampai tinta ini yang ada di dalam Petri ini menyebar dengan sempurna ke setiap bagian dari cawan Petri. This is diffusion. So, do you understand, everyone? Any question? Is there any question? Is there any question? What I did just now, this one, is point 3 of what you will include in your today's assignment. Okay, point 1 is the definitions, which already discussed before. Point 2 are the examples. Point 3 is how you are measuring the diffusion, observing diffusion. Point 4 is how you are observing and studying osmosis. Guys, things will be different if, for example, you change the temperature of the water, okay? Instead of using tap water, like what I am doing now, you use warm water, that could be different. If you use ice water, that could also be different. So you can include that in your short writing. I guess that's it. Any question? Again, I'm going to ask you, is there any question? Now I'm going to remove the spotlight, and I want to see if you really, how many of you understand my explanation so far? Please use the raise hand feature on your Zoom to tell me that you understand. Raise hand feature if you understand, please. Raise hand if you understand, please. Raise hand if you understand, please. Raise hand if you understand, please. 18, 19 participants, 20, 22. Raise hand if you understand, please. Raise hand if you understand, please. 24, 24, 24. Okay, that means 20, 25. Nice, 24, 23. Oh, okay. Okay, thank you very much, guys. I thank God because you understand. But really, is there any question? No question at all? Well, thank you. Remanus want to ask question? No? No? Okay, okay. So, guys, I won't hold you any longer here in this Zoom meeting. Yeah, actually, nobody asked me about how to do experiment related to osmosis, but it's okay. Remember that I'll be waiting for the submission of your project, collaboration project report today. And as for today's activity, today's report, today's simple writing, I wouldn't call it an essay, okay? It's just a simple writing which contain the 4 items I talked about before. Okay, I'll be waiting. You got 1 week to finish today's assignment. I guess that's it. If there is
Dan mau nanya,
yes, finally, go ahead, Mr. Ryzen.
kalau apa yang proyek kolaborasi satu orang aja atau semua kirim?
This is one of the FAQ, right? Frequently asked question. Guys, each and every one of you will submit a report. Okay, each and every one of you. So everybody will submit a report. Then you ask me question, but sir, we do this in teams, we do this in groups. Yes, I am aware of that. That's why, for example, if Mr. Ryson belongs to one group with, for example, Miss Jocelyn and Mr. Aaron, for example, okay, then the content of Mr. Ryson's report could be exactly the same with the content of Miss Jocelyn and Mr. Aaron's reports. Is that answered, Mr. Ryson?
Yes.
Thank you very much. So like that, everyone. So everyone have to submit. Everybody have to submit. Okay, everybody has to submit. Anybody else want to ask question? Go ahead. Thank you. Thank you too. You're welcome, Mr. Ryson. Anybody else want to ask question? Go ahead. No? Okay. Oh, Francisca want to ask question?
No, sir.
Oh, okay. Okay. Because you are still raising hand, so it's okay, no problem. Okay, everyone, and as you can see, until this very second, the ink hasn't been perfectly distributed, yeah, filling up the petri dish. I don't want you to wait. If there is no question, then I will allow you to leave. Thank you for coming. Stay healthy, stay safe, stay happy. God bless you all, guys.
Thank you,
Thank you,
Mr.
Mister.
Thank you very
Thank
much.
you, Miss. Thank you, Mister.
Thank you. Thank you very much, everyone.